I've been following this group of scientists from Italy that proposes super interesting theory. And as far as I know, they are the only ones working on this that suggests that the SARS-CoV-2 virus, the virus that started the COVID-19 pandemic, can infect bacteria inside our gut. And as a consequence, the bacteria inside our gut could act as a long-term reservoir for retaining virus in our body for long periods of time. And they recently published a paper delineating number of new experiments to prove their theory. I covered some of their work before in terms of the ability of the virus to infect the bacteria. So I covered their theory. But in my opinion, why I'm a big fan of this group of scientists and why I have been following them is because, as far as I know, and remember, this is only based on limited knowledge, personal knowledge, because I can only study a sliver of scientific information available. They are also the only ones who have ever shown or proven that vaccine spike protein can persist in the body for long periods of time. I covered that in a video as well. They were able to show that vaccine spike protein can be found in the blood in individuals for up to 6 months. Everyone else, whoever claims, oh, we suspect that this is spike protein from the vaccine or post-vaccine, they usually infer this. They don't prove it. They infer this on account that there was no signature of infection after vaccination, but it never discounts the possibility that the spike protein could arrive because of asymptomatic infection. So that's why I'm a big fan of these guys, because they do really cool, solid science, and they will use some of this technology in the science I will tell you about today as well. And I also had the opportunity to collaborate with the doctor that spearheads this group. In my mind, I'm not very good with names in general, but this is Dr. Carlo Brogna. And I had an opportunity to collaborate on one of the review articles with him as well through connecting scientists. We never met or anything like this. He didn't even know that I was pretty excited about that. But this is why, because I actually followed their science. Now, let me tell you about the work they did. They did 3 very cool experiments to show to prove basically how the SARS-CoV-2 virus could indeed infect bacteria. So they did electron microscopy, and I'll give you details on that, which in essence allows you to get detailed photographs of the virus, if you will, or the bacteria being infected by the virus. They also did what is referred to as mass spectrometry, which allows you to monitor what proteins are present in a sample. This is the type of technology that they used to show that vaccine spike protein was being identified in samples that they analyzed in the past. And in this work I will tell you about today, they're going to use that technology to show how new spike protein was being generated inside bacteria. And they also use next generation sequencing, which is a code for detection of genetic material of the virus. Well, it's any genetic material, but in this case, they use that technology to detect the genetic material of the virus. All right, so then let's break this down. This is a very cool paper. I liked it because it's visually stunning. So let's go through those electron microscopy images because they're quite spectacular. And they use 2 types of electron microscopies. When you hear electron microscopy, these are the most powerful microscopes you can think of, and they allow you to see images on atomic scale, basically. It's the most powerful magnification, as far as I know, that we can get. Maybe there's something more powerful that I don't know about, but as far as I know, this is the most powerful technology we have. And it gets down to basically molecular level. So there's 2 types of electron microscopies, and they both use beams of electrons. And the first one that we'll talk about is scanning electron microscopy, this is where you shine electrons onto your sample, and electrons will bounce off the material, and that's how you get an image. And this way, you obtain three-dimensional images of the sample you're interested in. Now, in order to do this, you need to do that by coating the sample with a very thin layer, usually of gold. So what did they do? They took a fecal sample, meaning from poop, of a person who was infected by the virus and had COVID-19, and they isolated bacteria and from the fecal sample. So this is why I like science, because only in science would you take poop bacteria and give it a bling for a photo shoot, right? So they specifically focus on two types of bacteria. And the reason why they focus on two types of bacteria is because these two bacteria have previously been shown to be dramatically reduced in people with COVID-19, which then imply to them perhaps the reason why that bacteria are reduced in COVID-19's patients with severe COVID-19 is because they are being destroyed by the virus. Why? Because if the virus infects the bacteria, afterwards, in order for the virus to spread, the cell that is being infected, in this case the bacteria, has to burst, and then you would produce new baby viruses. So then if you're infected and your gut bacteria is indeed being infected by the virus, then you could reduce the population of that particular bacterial species. Now let's see if I can recount the names of the bacteria, and I will purposely mispronounce them. One of them was something like Faecalibacterium prusnitzi. I'm not gonna get it right for sure. And the other one was something like Dore Fornici genera, something like this. Like, I knew I was not gonna get it, but I wanted to give it a try just for laughs. Either way, there's these two specific bacteria that they were focusing on, and that's the reason why. So what did they do is they took the fecal sample and they used it, basically checked it out under the electron microscope. They used that scanning electron microscopy technique first, and that basically means they coated the sample with gold, and they shine the electron beam, and it bounced off, and you see these spectacular images of bacteria that have these very weird bulges on them. And in 3 panels, you see progressively higher magnification of these bacteria. And then last panel, panel D, is a fecal sample from a healthy individual. So not a person who was infected and had COVID-19, but a healthy bacteria. And you can see its shape is intact, smooth, versus the bacteria from individuals who were infected by the virus. You see these very weird bulges, and they suggest that this could very well be the viruses being produced inside the bacteria, and you see those bulges there. All right, so that's the scanning electron microscopy. They did another one, which is called transmission electron microscopy. So scanning electron microscopy gave you the picture of the outside surface. Now let's focus on the second one. Transmission electron microscopy is something else. In essence, you take the sample and you cut it super, super thinly. If you take the hair, like one of my hair, and you cut it 1,000 times, so you get a 1,000th of the thickness of my hair, that's how thinly you slice the bacterial sample. So it's insanely, insanely thin. The reason why you do this is because you get an atomic wallpaper, and it's so skinny and so thin that this allows you to shine electrons through. And that gives you three-dimensional or two-dimensional view of what's inside. And in these images, again, they progressively increase the magnification, and you see once again these structures, these bulgy structures, and here the electrons, some of them pass through, some of them scatter, some of them will be absorbed. So like certain atoms will just seduce the electrons and will use its magnetic power to seduce electrons. And as a consequence, some of the areas of the image will look brighter, some of them will look darker. It gives you this illusion of three-dimensional image when it's actually two-dimensional, but it gives you a very nice sight of what's inside these bacteria. And you see, again, these bulgy parts, which they suggest might be indeed the virus. So, and then again, they have control samples from uninfected individual, and you don't see these additional structures that are being formed inside the bacteria. So very cool images. Now they went one step further and they took these images and they targeted with antibodies that would attach themselves to nucleocapsid protein. Now, nucleocapsid protein is one of the SARS-CoV-2 virus proteins. It's inside the virus. So they use antibodies that would recognize that viral protein. And then they use a second antibody that recognized that antibody. And that secondary antibody had a tiny amount of gold. Again, why gold? Because then when the electrons hit the gold surface, you just see a black dot. It's just dark, dark feature. And here now you see how the images, you see these tiny black dots. And every time now you see in this image where they use these antibodies to target viral proteins, every time you see that little black dot, that tells you where that viral nucleocapsid protein is. So they're clearly demonstrating that viral proteins are present inside the sample. Very, very cool. So that's set of experiments number one, where they use microscopy to show that infected bacteria have abnormal features that corresponds to viral infection, which they then proved with the antivirus protein antibodies. So it's same like we use antibodies to target spike protein, but instead of targeting spike protein, they targeted another antibody, another protein, which is that nucleocapsid protein. Now, in the second set of experiments, they use that mass spectrometry. So what is that? Remember, proteins are made out of amino acids. So amino acids are building blocks of proteins, and normally you link many, many amino acids together in tandem, and you make a very long chain. And because amino acids have charges on them, they will start interacting with one another, and as a consequence of these interactions, they start forming three-dimensional structures, and you end up having a three-dimensional protein or some sort of a molecular robot that does some function inside our cells. So there's obviously thousands of different types of proteins out there, and that's the makeup of proteins. Now you can cut them up. We have natural enzymes inside our body that can cut enzymes. We can do this in a lab as well. And you can cut up a protein into fragments. These fragments are called peptides. And even spike protein, the reason why SARS-CoV-2 virus is so infectious is because spike protein needs to be cut as well. And that's what helps the virus to be more infectious. So, but when you cut up a protein, such as say spike protein, into tiny fragments, these fragments are called peptides. And you can use this technique called mass spectrometry to analyze the makeup of these peptides. And this tells you the presence of a protein in a sample. And they used nitrogen atom, that a special type of nitrogen atom that is not commonly found in nature. It's a rare type of nitrogen that's heavier, and they added it to the sample, and they were able to show that as the virus was growing inside bacteria, and as a consequence was producing spike protein, this heavier nitrogen atom was incorporated into the spike protein. And they were able to show that along the growth in the amount of spike protein, the viral genetic material, RNA, which is the virus genetic material is made out of RNA, was also growing, and they correlated in the amount. So the more genetic material that they were observing in the fecal samples, the more spike protein was being produced, incorporating that heavier nitrogen atom. So that's another experiment that they were able to show that new spike protein is being produced inside the bacteria. Okay. So, and then the last experiment was the sequencing experiment. And this is also really neat. They took these two type of bacteria, they grew it in a culture, which is basically a liquid that has food for bacteria, and they plated it. They put it on some solid plate, and these bacteria grew like a solid carpet on top of these plates. And then they took those fecal samples from that infected individual and they put it on top of that bacteria. And because it had virus in it, the virus started infecting the bacteria on the plate and killing it as a consequence. Because again, remember, as the cell that produces the virus, eventually it bursts. It just literally gets destroyed. And as a consequence, that film of bacteria created a little hole, and these holes are referred to as plaques. So they were able to see these plaques, and they cut those plaques out, and they sequenced the genetic material out of those plaques. So they specifically focused on sequencing RNA, which is the genetic material made out of a virus. And for each type of these two bacteria, they were able to show 50 to 60% of entire SARS-CoV-2 virus genome. So again, they were able to prove that, look, these bacteria clearly are infected by the virus and are producing virus. But this is what's really, really cool. They were also able to show that depending on which bacteria was being infected, they produce different type of virus mutants. We know them as variants. You know, we keep hearing the variant this, variant that is being infecting us, and so on. Basically, variant is a polite word of saying mutants, and they different type of bacteria were producing different type of mutations. So these are the type of experiments they were able to show, to start showing that, look, we are not just being theoretical about it. It really does look like bacteria is indeed being infected by this virus. Very cool experiments. So they clearly showing that besides our own human cells, the virus could also be infecting bacteria inside our gut. It also means that these bacteria that are being infected in our gut could be producing different variants. They're saying that perhaps it's the bacteria that are more responsible for production of different variants than our own human cells, because the human cell's diversity is probably much smaller than the diversity of bacteria inside our gut, which we know has lots and lots of different types of bacteria. So it might be our gut that's responsible for producing the different variants as much more so than we would expect from our own human cells. And of course, this changes everything, and it changes a lot, because it means that the virus not only infects our own cells, but also gut bacteria. And that means that there's also the fecal transfer of transmission of the virus, potentially, not just from us breathing around. And what does that mean? They suggesting that this could mean few things for how we might have to handle this virus. And one of the things is that we could be infected by the virus. We now look healthy. The infection seems to be gone, but the virus could be sitting inside our gut and we don't even know. And perhaps the infection could come back. And it's not because we are being reinfected. It's because the virus that's sitting inside our gut could take opportunity of weakness of our immune system. Because let's say you got stressed out because your partner cooked a lousy dinner or something like that, and suddenly, boom, you're flaring up again. So completely different concept here that we are not considering. And look, as far as I know, and again, I'm not sure if this is correct, but I think they're the only ones who are studying this. And I actually really like, they mentioned in a paper that something to the extent like our results should be viewed as controversy and should be validated or verified by other people. So they're inviting other scientists, look, we should be studying this in detail. Let's start looking at this in greater detail. Don't just rely on us type of thing. We need further verification and replication of this data because it's too important. And we do have viruses that infect bacteria. These are called bacteriophages, but coronaviruses are not recognized as bacteriophages, or they're not recognized to have bacteriophage-like behavior. And these guys are suggesting that could be it. And there is some supporting evidence for this. For example, they mentioned, look, for long COVID individuals, it is often, their symptoms are often accompanied by gastrointestinal issues, right? Also, the nucleocapsid protein has been shown in the past to persist for a long time inside guts of people with long COVID. Also, the amount of one of these bacteria that I attempted to tell you the name, that Faecalibacterium prushinsky, that one of those, Prushnitzi, one of those bacteria inversely correlates with the severity of the disease, meaning the less you have of this specific bacteria in your gut, the more likely you're going to have severe COVID-19. And one more that they mention is, look, we also have heard about this sewer water be acting as a signal of impending waves of infection. And now and this was a bit of always a mystery. And now this might be explained as to why that is because, well, more people are getting infected, more people are spreading the virus via poop bacteria, and it ends up being in the water. And eventually, the more you see it, clearly it means the more people are already getting infected, and boom, you get a wave that is later on accompanied by people, say, going to hospitals or something like that, right? So in essence, the gut could be infected by the virus. So you could have a giant virus party inside your gut, but hey, clearly the more rowdy that party gets inside your gut, the more severe the symptoms of COVID-19 could be. So something we should definitely be looking at greater detail. And I just wanted to share this because I love the type of work and the type of images these guys are getting. And also it would completely change of how we should be dealing with the virus and infections, and also how we should be attempting to stop transmission as well. And in one of the past videos, we did discuss that they attempted to treat people with antibiotics to help deal with more severe outcomes. All right, I'm gonna wrap it up here, and please hit a like button if you enjoyed this topic. Please subscribe if you want to hear more crazy information about science with awesome photographs from cool scientists around the world. And definitely more about gut microbiome. This is a topic of interest of me. I'll be doing, I'll be doing more videos on this as well in general, because look, your gut is super paramount to your general health. And it's also extremely powerful precisely because of the bacteria that are inside your gut and how these bacteria, which is referred to as microbiome, can influence your entire body. It's just, there's too much of it and it's too powerful. And to some degree, how your gut goes can also determine how your general health goes. So keep that in mind. And last but not least, please check out my Patreon account as well. I always say now, I first screen all my videos on Patreon account, and then my supporting members vote as to which videos get to be displayed on YouTube. Some of my Patreon account videos are free, some of them are not. All right, so you don't have to be financially supporting Patreon to still get the free content as well. So check that out. And I'll see you guys in another fecal installment. Bye, everyone.